Hello everyone. So let's today discuss the topics osmosis, imbibition and plasmolysis. So you are all familiar about this. You may have studied in these topics in your school levels also. So first of all, let's see what is imbibition. Imbibition means the water absorption by a dry cell. Okay. Suppose we take water in a beaker and then put a dry cell. Suppose for example a gram seed. Okay. Then when we put the dry cell in the water solution, then what happens? The water is absorbed by the dry cell and it gets swelled, isn't it? That swelling is known as the imbibition due to absorption of water. Now the water which is entering into the cell is called the imbibate, and the dry cell which is absorbing the water and swelling is known as the imbibate. Okay. Now the plants, they are actually imbibants are normally hydrophilic. That means they are water loving. That's why they very easily absorb the water. Now this actually plays a very important role in water absorbing or the transport of water mechanism. Got it? Now the next is osmosis. Osmosis means the movement of solvent from its higher concentration to its lower concentration through a semi-permeable membrane. Now we know that semi-permeable membrane means the membrane which allows only the movement of solvent, isn't it? Not solutes. So through that membrane the movement of solvent will take place from its higher concentration to lower concentration. Now in this diagram you can see that it is a solution which is separated by semi-permeable membrane. Now this is a low concentrated solution, that is a high concentrated solution. That means solution A contains 9% of water, 1% of sugar, and the solution B has 9 sugar, 1 water. So definitely the concentration of water is higher in case of solution A. So the water will move from solution A to solution B, that is through the semi-permeable membrane, and that particular process is called the osmosis. Now, when actually water enters into the cell, then what happens? A pressure is being created. That particular pressure is known as the osmotic pressure. Now, osmosis is of two types. Endosmosis, in which the water enters into a cell, and exosmosis, in which the water comes out of the cell. Now, for example, if we take a resin and dip it in water, then what happens? The resin swells, it absorbs the water. That means the cell membrane will act as a semi-permeable membrane, and that particular cell, what happened? The water enters into the cell. So that is endosmosis take place. But if we put a grape in a sugar solution, then what happened? The concentration of water or solvent in the grape is more than the surrounding solution. So exosmosis will take place. That means the water from the cell grapes will come outside. Okay. So these are the two examples of endosmosis and exosmosis. Now sometimes what happens actually due to some particular things. Okay. Now for example, reverse osmosis may take place. That means the expulsion of pure water from a solution through a semi-permeable membrane. Okay. So that particular thing which we observe in normally case of removing salt from saline water, that is called reverse osmosis. Now while studying osmotic concentration, actually we find three different types of solutions: a hypotonic solution, hypertonic solution, isotonic solution. In case of hypotonic solution, the solution has lower osmotic concentration as compared to other solutions. Hypotonic solution, it has higher osmotic concentration, and isotonic solution, both the solutions have equal concentration. Okay. Okay. Now the third topic is the plasmolysis. Plasmolysis means the shrinkage of protoplast. Okay. So how the protoplast will shrink? It will take place when a cell is dipped in a concentrated solution. Then what happens? Exosmosis will take place. That means the water from the cell will come out to the solution, surrounding solution. Then what happens? When due to loss of water, gradually the protoplast shrinks, and that shrinkage is known as plasmolysis. Got it? Again, what happens if we put this plasmolyzed cell in water? Then what happened? The shape regains. The protoplast takes its original shape. Actually the cell wall being rigid, it doesn't change its shape. but the protoplast, it's being elastic and flexible structure, so it shrinks easily, but whenever we dip in water, it regains its structure. That particular phenomenon is known as deplasmolysis. So these are three important processes related to the water transport, okay, or substance transport. So osmosis, imbibition and plasmolysis. Go through them properly, and if you have any doubts, then please let me know.